We may do this pretty informally, with only one person here live and one. Let me ask you, your name is? And what sort of background do you have?
I'm a second-year student, PhD student in immunology.
Oh, okay. So you're in GBS Immunology?
Yes.
Your second year, so did you take, well, you probably didn't take then the genetics module or the bioinformatics?
No, I didn't.
Okay. And do you have any background in bioinformatics or genomics?
No, I have no background in bioinformatics.
And how, what's your background in genomics or bioinformatics?
No background in genomics at all. I'm a systems analyst at Children's of Alabama.
Okay, so I mean, I'm gonna. It's hard for me to switch out of a more formal presentation mode, but let's just stop this. Let me get these things off the screen somehow.
Let me introduce you to our. You can introduce yourself.
So yeah, I'll just introduce myself. So I am a professor in the Department of Microbiology. I'm also director of Informatics for the Center for Clinical and Translational Sciences. And so what I do is support a number of bioinformaticians who provide bioinformatic analytical services to people on campus. I'll show you, I actually have a slide with their names on it and such coming up. So, you know, the idea here in this talk was just to kind of give you an overview of what modern day genomics is, the type of data that we collect, and sort of how we approach analyzing that data. So just showing some of the examples of the data that's collected and how it's analyzed. Okay, let's see if I can get the slides to change themselves. Okay. You know, did you pass out any of the information that I had given you last year or made that available?
No, that becomes available at.
Okay. So either we'll have this presentation, but I think you also have maybe a Word file that has bibliography. It's in here anyway.
It's the same, I don't remember if you gave me this year, but the materials are the same from last year.
Yeah, it's the same from last year.
I'll send out everything.
The presentation's changed a little bit, but otherwise it's the same.
You send me the new presentation, I'll get the new presentation, everything else.
Okay. Now these are just different websites that have different analytical tools that are available. The easiest thing is really to just bring them up and see what type of information they have. I'll touch on some of these as I go along. And then there are a number of journals that publish articles that provide you with links to different resources. There's a database issue that's part of nucleic acids research every year, and it talks about available databases. Then there's a web server issue, and those are just web-based analytical services. So you can connect to them online and get access to a variety of tools. Then the other three, Bioinformatics, BMC Bioinformatics Plus, are just a few of the journals that have as specialties bioinformatics research. Probably you wouldn't want to go, for the most part, to the specialty journals. If you're looking for a way to do something, nucleic acids research or some of the other links I just provided before would be better sources of information, unless you're actually interested in the bioinformatics research that's ongoing. These are the list of review articles that talk about the use of genomics and next-generation sequencing for both research but also in the clinical setting. And so these are just a few of the ones you can check out. So at UAB, as I said, we have the CCTS that provides informatics services. I also am director of the bioinformatics services for the AIDS Center, but then the Heflin Center for Genetics, the School of Public Health, Section on Statistical Genetics, both provide bioinformatics services that are available. And then we work closely with the group in UAB IT that provides the high-performance computing infrastructure that's needed to analyze all this data. So in CCTS informatics, people can come to us with, in essence, any biological analytical problem, and either we'll help with that ourselves analyzing it, or if we think it's more specialized and there might be someone else on campus that can help analyze it, we'll direct you to the right person to go to. But bioinformatics analysis, next generation sequence, we do a lot of microbiome analysis. I'll talk about some of these in brief. And as I said, we have 5 bioinformaticians who are available and several programmers who are available to provide the support. This is just the contact information. We do provide, it's nice to have available some of these all-inclusive packages that give you access to different bioinformatic tools. They're either standalone packages that run on a PC or a Mac. There are a variety of those available. They tend to be rather expensive. We do provide something called Galaxy here, and that provides access to a large number of analytical tools. There's a learning curve there. So we offer lectures in a variety of courses or a workshop, and you sort of email me or keep an eye out if you're interested. But we can also give one-on-one assistance. But Galaxy, all you have to do is log on to it. You log on here at galaxy.uabgrid.uab.edu. It'll prompt you for your Blazor ID, so you click on UAB and you put in your Blazor ID, and then you get access to this web page. And then over here on the left shows you some of the categories of tools that are available. It allows you to do workflows. That is, it allows you to repeat the same set of different programs in the exact same way repeatedly on different types or different sets of data. And so that allows you to automate a lot of the analysis that you might have. Okay. Here. Okay, so what is next-gen sequencing data? So I don't know if you've had in your courses a little bit about sequencing data. Obviously, what you're trying to do is interrogate the cell, interrogate the biological system, and learn as much as you can about that system. The advantage of next-gen sequencing data is that it really gives you access to a lot of different points, a lot of different sources of information that are in the cell. It's kind of amazing how much you can do just with sequencing information, how much you can interrogate the processes that are ongoing in the cell. So you can look at the genomic sequence, you can look at expression of genes via RNA, seed. You can look at epigenetic markers through methylation or through binding of transcription factors to DNA. All of these you can look at via doing some form of sequencing with next-gen sequencing protocols. Then there's a lot of other data that's considered in the context of biological big data. What it gives you is a whole hodgepodge of stuff that you can't make any sense of. So really the idea of bioinformatics is to help you make sense of it. So you use the computer to help you analyze that information. The bioinformatics is really fairly simple when you boil it down, because all you're doing is looking for patterns. So you want to find a pattern in some piece of data, whether it be in next-gen sequence data, whether it be in expression data of different genes, but you look for a pattern that's characteristic of your system, and then you see if that pattern occurs in other systems. And if you see the same pattern occurring in different places, that suggests there's something in common between the biology of the two different data sets, of the two different maybe experimental groups that you're looking at. And then if you find that this pattern is always associated with a certain phenotype, maybe it's a disease, then if you see the pattern again, that then allows you to make an inference that that disease is present in that next data set. And so that's all bioinformatics is, identifying the patterns and then looking for repeated occurrences of the patterns. Sequencing, as I said, is a major way these days of identifying patterns. So you do sequencing, you play a lot of little tricks in terms of how you generate that sequence, and then when you analyze it properly, you can identify patterns that mean all sorts of different things. These are just a series of slides to show how sequencing technology has evolved over the years. We used to use radioactive labeled nucleotides that run on these big slab gels. You had to read these by hand and interpret them yourself, and you had to write down all what the bases were. And at most, you could do a sequence of maybe 500 bases, and you had maybe a dozen, 2 dozen lanes at most. So you're kind of limited in what you could do. It took a lot of time. Then they automated it by using fluorescently labeled nucleotides, and then a laser could automatically read the bases. So you didn't have to read them by hand yourself, and then you still had the same slab gel and the limitations on the number of lanes. It was improved again using these capillaries. So now you can use a capillary of up to 96 capillaries for some of these big sequencers. That's how the human genome was originally sequenced. But you can still only sequence 96 samples at one time. That is, you can only sequence 96 individual strands of DNA, and at most get, again, maybe 500, maybe a few hundred more bases, but that's about it. So 500 times 500 bases times 100 lanes of about 100 capillaries. So, you know, that only gives you a very limited number of sequences and sequence information that you can generate. And so that was the basic limitation. But then came along what we call the next generation sequence. This is Illumina. There are others that are also used. But now what you have a chip like this, and you put your samples on this chip. And on the chip, you can put, in essence, millions of separate sequences of DNA on the chip, and every sequence is read. That is, every sequence is sequenced. We determine the sequence of every single one on that chip. So instead of only sequencing 96 samples at one time, in essence, we're sequencing millions of different reads, millions of different pieces of DNA all at one time. And by using barcodes and playing lots of other tricks, you can sequence multiple samples at once. You can sequence upwards of 100 or more different samples, each of which contains lots of different pieces of DNA in it. So we went from a very limited capacity for sequencing all at once in this new methodology to be able to sequence the millions of bases. And this just shows that the change that happened. So you would sequence less than 0.1 megabase, that's 0.1 millions of bases, so 100,000 bases at once using the old Sanger technology. These days, you can sequence 1,000 gigabases, or 1 terabyte, so 1 trillion bases all at once in one particular run.
Elliot?
Yeah.
That's about 10 years old now, right?
It is about 10 years old now, right?
Are there any newer improvements?
So yes, so in 2 ways. So one, the next-gen sequencing technology itself, when that came into being, that was a revolution.
I mean,
It slowly evolved.
Generation.
So
Yeah.
that next generation has evolved, so it's much more efficient and faster, and you can get longer reads than you did originally. But there are lots of other newer, different sequencing technologies becoming available. And they're geared at giving you longer sequences of the same strand of DNA. And that overcomes some of the problems that we have now. As I'll show, when we assemble a sequence now from individual reads, you have to overlap lots of individual reads and combine them into one single long sequence read. And you aren't guaranteed that they all came from the same original strand. The newer technologies guarantee that. And so that will be the next revolution. They're still highly error prone, so the new technologies, you have to have a lot more redundancy than you do now in order to ensure that you don't have the errors creeping in. You saw this before. Jake Chen showed how the databases have grown due to the advent of next generation sequencing technology. But the thing that I emphasized before that I will again is that you can do lots of different things with it. You can look for variation in the whole genomic sequence of an individual or a model organism like a mouse. You can just look at the coding regions in those sequences. You can look at expression of genes through transcriptomics or RNA-seq. We do microbiome analysis that I'll talk about later. I don't think I'll answer that. And other techniques, epigenetics, by looking at methylation patterns or looking at transcription factor binding sites. All of that is done with the same basic technology. You just switch it up a little bit here and there. But the key, of course, is analyzing all this data. So that's where the bioinformatics comes in. Pretty much, you start at the same place, no matter what technology you're trying to use, as long as it's a next-generation sequence technology. You have to assemble the sequences. You have to make sense of the sequences. So imagine you have millions of small little runs of sequence. So each sequence read in one of those strands is maybe 100 to 150 to 300 bases long. But you have millions of those. And so if you're sequencing a complete human genome, you're going to have millions, actually billions of those individual reads. And what you can do, you can assemble all of them into one single long sequence. And in order to sync, and when you first start off, if you don't know what it is, how they assemble, you have to compare every read to every other read, which is a computationally pretty lengthy process. But you compare every read to every other read, and then you order them according to how they line up with each other, what sequence they have in common, and this allows you to build up progressively a complete sequence of whatever it is you're studying. And so that's how they derived the human genome originally. They did lots of sequencing of the human genome, cut into all sorts of little pieces, and then put it back together again when they actually have the sequence. It becomes a lot easier now if you have a reference genome. So now we have a human reference genome. So all we have to do is take every sequence read. We don't compare it to every other sequence read. We just compare it to the single reference, in this case the human reference genome. And that allows you to line it up very quickly against the reference genome. So that makes a big difference. It makes it a lot easier. So what do you do with that information? Well, one thing that you do is look for variants. So you have a reference genome, and you ask the question, how is this genomic sequence from my study different from that of the reference genome? Let's say you're studying some autoimmune disease like arthritis, and you have a hypothesis that there may be particular variants in the human genome that map to the phenotype of arthritis. So what you do, you compare the sequence of arthritis patients to normal control patients, and then you look for the variation. And so these are the sequence reads. They're aligned against the reference genome, perhaps the human genome, and then you look for areas where the sequence reads differ from the reference genome. And then you try to map those to individual genes and use programs to decide how important they are. This just shows you the data itself. So each of these is an individual read, and each of these colored bars is where that read differs from the reference. This is sort of mapped to the reference here. And then this shows the gene, the exon-intron structure of a particular gene in this case. So any of these variants that occur within this gene would map to this area or this exon here or this one here. Now what you don't see is consistent variation across all the reads. And that's what one would expect if you actually had a true variant. But you look here, here's a case where here we have a variant, and you see about half of them are A, I think red is A, I forget. But about half of them are one base. All the other half are the reference base. So this is a heterozygous position. So half it looks like the reference, half is a variant in comparison to the reference. Here, you see everything differs. And so, well, I take that back. Here we have like 60%, 37% G and T. So this also is probably a heterozygous position. But these are the positions you look for, and then importantly, you look for them occurring, at least in this case, in the exon, which may mean that it causes a coding sequence difference, maybe therefore a protein difference, maybe therefore a phenotypic difference that could result in disease. But then what you really do is you take note of all the variants you see in a study. So you want to design a study, in which you're studying arthritis. You have a set of control patients. You have a set of experimental patients who have arthritis. You do genetic sequencing on all of them, and then you look for genetic variants that pop up that are present only in the arthritis patients but not the control patients. And so this shows individual variants that are present in these populations. This shows the number of times they occur in cases, that is the experimental group. This shows the number of times they occur in the control group. And then so you look for variants that are more heavily present in this group here. So this first one might be significant because it has 9 in the case group, only 1 in the control group. And then you look further and you try to map all of these across the whole human genome. And so each one of these dots represents a variant, and each one of these colored bands represents a different chromosome. So you look for peaks occurring here. And a peak may indicate the same variant in the same position in the same gene. Or it could be the same gene has different variants in it, but they all affect the phenotype. So you may have a gene that a variant that interrupts a particular gene, and there could be another variant downstream that also interrupts that gene. But they both may cause the same phenotype, that is arthritis in this case. So you look for a peak that coincides with a particular gene in your experimental group. And this is just another way of sort of visualizing that.
But it's chromosome 6.
So that's right. So in this case, so this represents all of chromosome 6, which is going to be several megabases in length. So, you know, there's a lot of genetic information there, but we can zoom in on that. And it looks like all of these may be present in approximately the same location. Now, actually, we could really be talking about hundreds of thousands of bases here, but when you zoom in, you'll find out whether or not all of these are present in or near the same gene.
Could it be the human polyploid antigen, HLA?
That's certainly possible. There are associations between
Can you
that.
repeat the question for the recording?
Sorry. So the question is, well, we have this band that appears in chromosome 6, and so what might it be? Now, in some cases, the question was, could it be HLA antigens that a particular variant is mapping to? We know that certain HLA types are associated with certain diseases, so yes, that's one possibility. Another thing you have to take into account when looking for significant human variation is that different ethnic populations will have particular variants that are only contained within that ethnic population. So you have to filter those out. You have to control for those ethnic variations. And there may be other types of variations you need to control for to look for the significant variations that might be directly associated with or causative of disease. So gene expression analysis, transcriptomics. So not only is the sequence of a gene important, but the rate at which or the extent to which any particular gene is transcribed into messenger RNA and then translated into protein. And so transcriptomics, we're looking as a surrogate for protein expression, we're looking at messenger RNA expression. And so what you do is you don't sequence all the DNA. What you do is sequence the RNA that's present in the cell, and the amount of RNA, that is the amount of sequence that you get back from one particular gene, is an indication of how much expression you're getting from that gene. So a gene that's only transcribed at a low rate will have a low amount of sequence associated with it. A gene that's transcribed at a high rate, you'll see many more sequences come out when you do the next generation sequencing. And so here are the exons present for a particular gene, and the read depth are the number of sequence reads that map to positions within the exon of the gene. That's what you're seeing here. Each of these is mapping to the exon of that gene, and so you can compare the read depth for all of these exons sort of combined in that gene to the read depth of other genes, and that shows you the differences in expression levels between genes.
So this is in one single cell, or is it?
So it depends. So normally what you're looking at is probably a tissue. It could be blood. Maybe you take a biopsy of a cancer tumor or a lung biopsy. So you're looking at expression in whatever sample, whatever tissue, whatever blood cells you've separated out.
In your opinion, would it be better to pull different subset of cells together, or
Frequently, it's not good to pool. I mean, it depends upon what your experiment is, what your hypothesis is. But remember, every different cell type may have a very different expression profile. And so when you mix them together, when you pool them, you're pooling the expression profiles, and you're losing all of that information that differentiates between one cell type or another cell type.
So can transcriptomics technology do single cell?
So yes, so the technology has improved to the extent that you can do single cell. Well, you can certainly do single cell sequencing to look at variation. I don't know offhand if you can do single cell transcriptomics or not. That may be pushing the limits of the technology. I might have to check that. But you can certainly do it on purified populations of cells. Then in addition to doing gene expression profiles in terms of the level of expression, you can look at alternative transcripts. So this shows sort of known exon-intron boundaries for a particular gene, and then here you see alternative transcripts that appear. And different transcripts may control different functions at different times and different cell types. So you can get a pattern of differential expression of transcripts from a single gene. You can do a lot of other things. I won't spend much time on this. A lot of people are interested in viral infections and whether and why you may see different phenotypes. That is, some people may have more severe disease when exposed to the virus. Other people have mild or inapparent disease. That could be due to the individual, their genetics, but it could be due to the genetics of the virus. So you can look at the viruses and see if you can't map specific genotypic changes in the virus to severity of disease. And so this is a virus, I think it's human cytomegalovirus, and again, you just do the genetic sequencing of your samples, and then you align it to a reference genome that's from GenBank, and then this just shows us where there are variations between the reference genome and the sequences of the viruses from the patients. And if we see particular sequence changes that always occur in patients with severe disease, that gives us something to study. That gives us something to follow up on. This is just sequencing of human cytomegalovirus, and I won't worry too much about that. We also do a lot of bacterial genomics, so the same thing. We're sequencing bacteria cells and asking the same question. Is there some property present in certain genotypes, certain lineages of bacteria that may make them more pathogenic than others? And so this is a study that was done with Mycoplasma, which is a small Mycoplasma Genomics, which is a small bacteria, and we derived the sequence working with a group at UAB of a little over a dozen different bacterial isolates, and then this just shows them all compared to each other, and other views of this will show the particular variants that are present in some of these strains of Mycoplasma, and you look to see again if they're associated with disease. One thing that's important a lot of times is you want to compare results from your system to other systems. So one thing might be useful is if you're studying human genetics and you're looking for ways in which the human genome controls expression of its genes, and maybe there are particular control elements that haven't yet been identified that can modulate expression of a particular gene. Well, if that's a control element that's important, maybe it's been conserved evolutionarily in other organisms similar to humans. And so you can take the human sequence around a particular gene and compare it to that of a monkey or a dog or a mouse, that's a possum, a chicken, a xenopus, a frog. And so this just shows the level of sequence similarity between the human and each of these model organisms. So you can see that this is a macaque primate, has the highest level of similarity to the human genome. And here, this again is the gene, the exon-neutron boundaries of that gene. So each one of these are the most highly conserved, the exons, between all these different organisms. These regions outside, just upstream or downstream of the gene, are highly conserved. These are potential regulatory regions. So these, where you see these conserved, may be places to look for regulatory sequences that are important in expression of this gene. So as I said, bioinformatics is really looking for patterns that can be matched up between different situations. Comparative analysis does just that. We're trying to match up patterns between humans and model organisms to tell us something about human physiology and human disease. These days, a big topic is microbiome analysis. That is, the microbial population that inhabits our bodies, every area of our bodies. They have an important role in maintaining health and also causing disease. So now we have techniques using next-gen sequencing again to really monitor that microbial population. So in microbiome analysis, so that's all that makes sense. Microbiome analysis sequences the 16S ribosomal RNA gene of the bacteria that inhabit our body. The 16S ribosomal gene is present in all prokaryotes, but it differs in certain regions from one organism to another organism. And so we can pull out the 16S gene by using primers that are shared between all prokaryotes, but then between those primers is unique sequence that characteristically identifies particular species of microbes. And so you use the common primers, and then you sequence everything in between those primers, and the sequence you get back will give you a profile of all the microbial organisms that were present in the samples that you were looking at. So again, you could compare samples between people with different diseases. Arthritis is a project we've looked at. So we look at patients who have arthritis and compare their microbial population in their gastrointestinal tract to control sets of patients and see if those microbes differ. And indeed, that's what you can see. These are just some of the ways of representing the data. Along here, what you see in this bar chart here is these are different samples, and then each of the colored bars represents a different microbe as identified here. And you can see how the pattern of this set of microbes here is distinctly different from the pattern of this set of samples here. These happen to be samples, I'll show you in a minute, that are derived from a study of Clostridium difficile infection. So Clostridium difficile causes severe gastrointestinal symptoms in patients. It can be treated by antibiotics, but frequently it keeps coming back and you can't get rid of it. So what they found is that an effective way to treat it is to actually have a fecal transplant. That is, you take a transplant from someone who has a normal microbial flora and then give it to the person who has the C. difficile infection. It repopulates their gastrointestinal tract, and it works extremely well, and they pretty much are cured in most cases. And so these are the donors here, and these are the recipients. So you can see how different the recipients are in this case from the donors. So you can ask the question, what happens to the recipients after transplantation? So that's a study that we did here. So this is what's called a principal components analysis, or beta diversity. So we compare all of the microbial population in one sample versus another sample versus another sample. And this shows you, it's actually in 3 dimensions, how different each sample is from every other sample. So when the samples cluster together, they have the similar microbial population to the other samples that cluster with them. So here we have their cluster pattern, then we color code them according to whether or not the sample comes from the donor, it's the recipient before transplantation, or the recipient after transplantation in the yellow. So you see the donors are over here. They're a little bit different from one another, but they're all present here. The recipients before transplantation are present here, so their microbial population is very different. Now, and this is only after 2 weeks for these and 4 weeks here, now after transplantation, you see they're beginning to move to look more like the donor profile here. So again, this is 2 weeks. The 4th week is looking even more like the donor. Here, this may be at a later time point. Oh, I'm sorry. This is actually transplanting these into mice, so you can do the same thing. You can take these samples here, put them into mice, and see if you can't recapitulate the phenotypes that we showed here.
So what is the goal for the people? transplant?
Sorry, what is?
What is the dose? How is they take it every day?
How is the fecal transplant performed? I think generally it's a single dose, and they can do it either with they can go in either direction and get it into their GI
How
tract.
often do they have to take the?
Usually just once.
Once a month?
No, just one time. So the transplantation consists of a single transplant, that is a single dose of donor fecal material into the recipient. That is enough to take. That is enough now to allow repopulation of the recipient's gastrointestinal tract with a new set of microbes, and in essence cure the C. difficile disease. And so we've been doing a lot of studies with this following these transplantation profiles. We've gone one step further. Instead of just sequencing the 16S gene from these bacterial populations, we can sequence everything, and that's what metagenomics is. So you sequence all the DNA that's present in the samples, and then you reconstruct the entire genome of the organisms that are present. That allows you to reconstruct some of the metabolic pathways that are present not in a single organism, but in a population of organisms. And that's what you do here, And what you can do here then is reconstruct these pathways. And so this shows you what enzymes and particular metabolic pathways are present and how much is present, in this case, in 4 different samples. So you see some of these enzymes aren't present at all in the sample we're looking at. Other enzymes are present in most of the samples, but for some there's differential expression. Here, like 3 has more of this particular enzyme here than others. And then you can reconstruct these pathways in totality by combining all of this different information. So now we can get an idea of why a particular microbial population might be associative or causative of a particular disease by looking at the metabolism that might be altered as a result of the presence of particular sets of microbes in an individual. Okay, so that's sort of, I've gone a lot faster than I normally do, but that really gives you a broad overview of genomics and how we utilize next generation sequencing, the type of data that we derive when we do the analysis. As I indicated, there are lectures that we provide that sort of are much more hands-on, that give students or postdocs or whomever access to the tools that you can use to analyze the data yourself. The GBS Bioinformatics, there's a GBS module in the spring for Bioinformatics, and you can sit in on that, and that provides a nice overview of different analytical techniques. There's a microbiome course that's offered in the fall that also goes into these techniques if you're interested in the microbiome. And there's several other courses, so those are posted, so keep an eye out. There are also workshops that are available. We're going to have a microbiome workshop in September that will go into some of the more detailed, involved analytical techniques that we utilize. That's the 14th and 15th of September that we have the microbiome course. So I'll just leave it there and see if you or Hope have any questions.